Hello, thank you for the opportunity to speak here today about neoantigen vaccines for personalized immunotherapy. My name is Asmann. I'm an associate professor of Biomedical Informatics at Mayo Clinic. Let me first give an overview of relevant biology and introduce some terminologies. First, the protein degradation pathway. At the middle of this figure to the left, proteins marked by ubiquitin tags are degraded by proteasomes, and the resulting peptides are transported into ER by TAP. In the ER, each peptide is loaded onto MHC class I molecules by many molecular chaperones. The peptide MHC class I complex is then transported to the cell surface for presentation to T cells. T cells then read each peptide to make sure it's not harmful. The question is, what are these MHC molecules that present peptides to immune cells? MHC are major histocompatibility complex proteins in vertebrates. In humans, MHCs are called human leukocyte antigens or HLAs. So HLA is the human's version of MHC. In this talk, MHC and HLA are used interchangeably. Human HLA genes are located on the P arm of chromosome 6. There are 3 classes of HLAs. Class 1, including HLA A, B, C, E, F, G. Class 2 of DP, DQ, DR, DM, and Class 3. Class 3 HLAs are not involved in peptide or antigen presentation, so we'll only focus on HLA 1 and 2 for this talk. MHC class 1 or HLA class 1 presents peptides from non-self proteins from within the cells to CD8 positive T cells. The non-self peptides could be from, for example, microbials, or in the context of cancer, mutated peptides. MHC class 2 presents peptide from extracellular non-self proteins to CD4 positive T cells through phagocytosis. As shown in the left figure, most cells, including tumor cells, express MHC1, and as shown in the figure to the right, immune cells, including antigen-presenting cells or APC, express both class 1 and class 2 MHCs, although only class 2 is illustrated in the figure. The APCs can pick up available antigens or engulf tumor cells. Now we're ready to discuss neoantigens. In the past decade, immune checkpoint blockade therapy has revolutionized cancer treatments. The immune system naturally recognizes and responds to cancer. However, tumors evade immune system, as shown in the figure to the right, by expressing cell surface immune checkpoint proteins such as PD-L1 and marking themselves as normal. ICB works by removing this camouflage. The efficacy of ICB remains limited and well below 20% today due to resistance or low efficacy of the ICB agents. Therefore, novel immunotherapy agents are being actively developed. Cancer neoantigen is one of the most promising next-generation agents that can improve ICB efficacy. Cancer produces different antigen. Neoantigens belongs to the subgroup of protein cancer antigens. Unlike microbials or self-proteins, neoantigens are from mutated protein from tumor somatic mutations that can be presented by MHC molecules to T cell receptors that consequently triggers immune response. This is the definition of cancer neoantigens today. Over the years, a cancer immunogram has been proposed around 7 classes of biomarkers that can be used to predict the ICB or immunotherapy treatment outcomes. As shown in this figure, from the top and clockwise are tumor foreignness as determined by tumor mutational burden. Second, the general immune status of the patient assessed from lymphocyte counts. Third, the tumor infiltration of tumor activating T cells. And the fourth, the expression of PD-L1 and the rest. Among these, the tumor mutations are the source of neoantigens. A brief overview how neoantigen works. The tumor cells, like the one in the center of this diagram, present antigens, neoantigens by class 1 MHC to CD8 positive T cells. In addition, antigen-presenting cells, such as the dendritic cells shown here as the large blue starfish-like ones, can also take up and present neoantigens by both class 1 and class 2 MHCs to CD8 and CD4 T cells, respectively. Together, this process leads to adaptive immune responses that kill cancer. This is how endogenous neoantigens work. However, endogenous neoantigens levels are often too low. So if we could identify what neoantigens a patient's tumor has and supply the extra dose by vaccine or other means, this exogenous the immune system to look for tumor. The COVID vaccines work the same way. And when ICV removes the tumor surface camouflage, the trained immune cell can readily target the tumor. As I mentioned, neoantigens come from somatic mutations, including single nucleotide changes and small insertions and deletions in genomic DNA. Also, aberrant splicing isoforms of the RNA transcripts and the genomic rearrangements, such as translocations, inversions, and copy number alterations that lead to gene fusions. It's worth mentioning that not all neoantigens are the same. Some only harbor a single amino acid change. Others, due to protein fusions or translational frame shifting, can be very different or entirely new compared to wild-type proteins. The latter are generally more immunogenic. Later, I will describe some of the algorithms we developed in-house to identify these potent neoantigens. The features that constitute two high-quality neoantigens from left to right, top to bottom include the high degree of non-selfness. For example, as I just mentioned, neoepitopes from frame-shifting indels have higher degree of non-selfness compared to peptides with a single amino acid change. 2, tumors are known to have immune selections during proliferation and progression and lose clonal neoantigens in new subclones. So if a neoantigen is from a passenger mutations of gene critical for tumor survival, it is more resistant to neoantigen loss. Third, neoantigens that can be presented by multiple HLAs are of high quality. And fourth, genes harboring neoantigen mutations is in stable genomic regions with lower likelihood of copy number loss will produce stable neoantigens. And finally, the studies also show that a minimal cell fraction is required to trigger immune response. So neoantigens from a small subclone may not be as effective. Oh, and finally, neoantigens need to be expressed at moderate or high levels. A few findings from previous studies I like to mention. An early study in melanoma showed that absolute majority of the mutations are not shared between patients at meaningful frequencies and can be considered patient-specific or private. Therefore, absolutely majority of the neoantigens are private. As shown in the figure to the right and the upper panel, only less than 0.005% of neoantigens are shared among 20,000 melanoma samples. Second, most neoantigens are from non-driver passenger mutations. From this melanoma study, as shown in the lower panel of the figure, only 8% of the class 1 neoantigens are from driver mutations in oncogenes, and all class 2 neoantigens are from passenger mutations. You may ask, melanomas are from random mutations due to UV exposure. Of course, the resulting neoantigens are random, therefore not shared between patients. What about other cancer? The answer is all very similar. For example, we studied about 850 breast cancer patients. 99.75% of the neoantigens were only observed in either 1 or less than 1% of patients. And towards the top, only 18 neoantigens were shared by more than 5% of patients. I can tell you that results are similar for other cancer types as well. Because of this, neoantigen-directed immunotherapy needs to be highly personalized as well. Neoantigens can be used to treat cancer in the formats of vaccines, cellular therapies in combination with ICV. In addition, neoantigens are also biomarkers that predict ICV outcome and patient survival. With the lack of accurate measurement of neoantigen load, however, tumor mutational burden, or TMB, has been used as a surrogate. But I want to show you later that the neoantigen load is better biomarker than this surrogate. This 2015 Science paper grouped the tumor's ability to produce neoantigens based on tumor mutational load, as shown in the y-axis. The cancers in green boxes in the green box with higher TMB, such as melanoma and lung cancers, are predicted to frequently form neoantigens, while tumors with low tumor mutational burdens to the left, such as some of the hemo malignancies, only occasionally form neoantigens. Here, I highlighted breast cancer pointed to with a red arrow, which has medium level of TMB, to show you later that TMB under represents neoantigen load and is not a very accurate biomarker. TMB is calculated as number of protein-altering mutations per megabase of coding DNA, which comprises of SNV, indel, and splice site mutations. First, the ratio of mutations versus neoantigens are often 1 to many. Second, both genomic rearrangements and RNA de novo splicings are not counted towards TMB. An early Nature Genetics paper showed that cancers in ovary or breast are C-class, most driven by genomic rearrangements but not small mutations. Our own work to the right showed that not all splicing events had associated DNA mutations. For 16 lung cancer patients with known MET gene exon 14 skipping events, 9 came from de novo RNA splicings. Only 6 resulted from DNA splice site mutations. Therefore, TMB underrepresents neoantigen load and is not a very good surrogate. This Nature Genetics paper from 2019 showed that higher TMB predicts better overall survival after ICV therapy in multiple cancers, highlighted in red boxes, but not in breast cancer, pointed to by the red arrow. However, last year, we published that the lower mutational burden, not TMB, is a biomarker for worse overall survival in breast cancer patients, independent of other clinical predictors, such as tumor infiltrating lymphocytes, breast cancer subtypes, tumor stage and age, and a diagnosis. Again, supporting the notion that neoantigen load is a more accurate biomarker. As shown towards the bottom of the slide, neoantigen peptides can be directly identified by chromatography and mass spectrometry analysis of MHC ligands. However, this process can be laborious and requires large amount of tissue. Today, neoantigen detection is mostly done by bioinformatics analysis of tumor DNA and RNA sequencing in 3 steps. First, patient-specific MHC is genotyped. Second, potential new epitopes from mutated proteins that are expressed in tumor cells are generated, and then bindings between MHC and the neoepitopes are predicted. At Mayo, we developed an in-house bioinformatics framework called RealNeo. Here is a diagram of the version 2 of the pipeline. So why are we developing our own methods while there are multiple published tools? Because neoantigen identification is a fast developing research topic with no gold standard. Furthermore, a state-of-the-art in-house pipeline will enable us to quickly incorporate new knowledge, add new functionalities, and performs QC and prioritization of the neoantigens without dealing with the black box problem. I don't have time to go into all the details, but we'll discuss 3 components of RealNeo, namely the HLA genotyping, identification of the neoantigens arising from aberrant RNA splicings, and MHC neoepitope binding prediction. First, the HLA genotyping. Each individual has different HLA genotypes. The first step of neoantigen discovery is patient-specific HLA genotyping from tumor DNA and RNA sequencing data. There are at least 20 existing HLA typing tools. Some can type both class 1 and class 2 HLAs. Others can only do class 1 typing. To benchmark and evaluate these tools, we look at the consistency of HLA genotypes between repeated sequencing samples of the same tumor with different sequencing types and depths. As shown in the 2 figures in the middle, the x-axis are the different patients, y-axis is the degree of consistency between repeated samples. OptiType, the upper panel in green, was the best class 1 HLA typing tool, and HLA HD, lower panel in green, was the best class 2 typing tool. I want to draw your attention to the big difference of performance between tools, so tool selection is a key. Second, the uncertainty of predicting HLA neoantigen binding. To the right is the correlation coefficient matrix of neoantigen binding affinities to class 1 HLAs between 8 published methods. The concordance is very poor. So our current strategy is to only study the neoantigens with consensus prediction among multiple tools. In addition, we are developing novel machine learning-based methods to predict the non-selfness score based on amino acid features such as hydrophilic or hydrophobic scores and dissimilarity between neoepitopes and the wild-type proteins, among other attributes. This is necessary because reports have shown that relying on binding affinities will lead to high false negative rate of about 30%. We need a neoantigen prediction tool beyond evaluating binding. Third, the identification of aberrant splicing neoantigens, which is a novel functionality specific to RealNeo. First of all, there is a category of DNA mutations located in splice sites that can lead to aberrant splicings, as shown in the figure to the left. The quantity of these mutations are on the par with indels and gene fusions, using TCGA breast, liver, and the 2 lung cancer types as examples. However, these splice site mutations are not the only cause of aberrant splicing, as previously shown in the figure to the right in the example of MET exon 14 skipping. De novo RNA splicing is responsible for majority of the aberrant splicing events. RealNeo is capable of detecting both categories of the highly immunogenic aberrant splicing neoantigens. Here is the algorithm for neoantigens from splice site mutations. As shown in these figures from top left, we start from DNA mutations and predict splicing donor or acceptor sites gain and losses using a published tool called Splice AI. After survey of close to 12,000 TCGA cancer cases, 13 different profiles were detected. Some are very frequent and others are more rare. The top 7 profiles represent 95% of the splicing events. The simplest profile, pointed to by the red arrow, and also shown at the figure at the bottom left, has a single loss of the original splicing site, acceptor site. This simple profile, however, generated rather complicated 3 different splicing alterations, depending on the location of the acceptor loss, either in the middle exon or in the last exon of the gene. It could lead to intron retention or exon skipping. Dr. Colton McNiven in our team painstakingly derived all potential splicing alterations from all profiles. Most are way more complicated than the simple example. Once we computed these putative splicing isoforms, we confirm that these splicing products are expressed by quantifying the expression, as shown by the y-axis of the figure to the bottom right, and by calculating a z-score, as shown by the x-axis, using the entire TCGA cohort as the reference, assuming individual specific aberrant splicing events are rare. Here is a summary of the aberrant splicing events and the neoantigens from splice site mutations. To the left are the counts of different splice site changes. For example, the combination of acceptor loss and donor loss, acceptor loss and donor loss in pink towards the bottom, that gave rise to splice site predicted splicing events. The confirmed predictions by both expression and z-score, and the number of neoantigen candidates in TCGA breast, liver, lung, adrenal, and squamous cell carcinoma. To the right are the counts of different types of splicing events, such as in-frame or frame-shifting exon skipping. I'll let you stare at it for a couple of seconds. Next, here is how we detected de novo splicing. I won't go into the comparisons between our approach and the literature approaches. I'll just say that our approach can detect multi-exon skipping events, which are novel. To the right from the top, we started with the junction support reads. For example, in a sample, we might detect both E1, E4, and the E1, E5 junctions. Next, we use a z-score threshold, assuming these events are rare. And it, in addition, require that the novel transcripts are expressed and the fraction of the novel transcripts, novel junctions, are at least 10%. Next, we eliminate E1, E5 if there are evidence showing the so-called E1, E5 junction reads actually also support E1, E4. Then we do another round of transcript quantification to make sure that the novel splicing events are indeed expressed. The novel transcripts will then be translated into k-mers for HLA binding prediction to identify neoantigen candidates. As shown in the figure to the right, the numbers here confirm that about 40 to 50% of the aberrant splicing events are detected by both approaches towards the top. And the majority of the splicing events are de novo towards the bottom. There are also a small percentage, around 5%, of the splicing events that are only detected by the splice site mutation route in the middle. The figure to the left showed that our two approaches detected common and unique types of splicing events. Last, I want to point out that because all neoantigen binding prediction tools use existing laboratory binding assays to train their model, the prediction confidence are not the same for all HLAs. For HLAs with large training data set, the prediction confidence are higher. As shown here, the class 1 HLAs are in red and class 2 in blue. Two class 1 HLAs, HLA A0201 and 0301, have the largest number of epitope binding assays available for model training. For class 2, HLA DRB10101 has the largest training data set. So for now, we can only confidently predict neoantigens binding to very few HLAs. Luckily, as shown in the right inlaid figure, the HLA with the most training data set are also the most prevalent in population. For example, HLA A0201 is expressed in more than 40% of the human population. So even though we only predict neoantigens binding to a few HLAs, we can still do so for the majority of the patients. I'm happy to report that RealNeo is highly accurate in predicting neoantigen HLA binding. Here I showed some results of novel neoantigens from gene fusion and aberrant RNA splicings. This is the laboratory validation results. The first bar to the left is the positive control, which is a peptide from the flu virus. The next two bars are negative controls. The validation rate was 100% for neoantigens that binds to HLA A0201. Some even bound stronger than the positive control. In the next few slides, I will focus on neoantigen vaccines as the next generation immunotherapy agent. Now that you have some confidence in our bioinformatics platform to identify patient-specific neoantigens from tumor genomic sequencing data, neoantigen vaccines can be delivered in the forms of peptide, DNA, or RNA vaccines, or as dendritic cell vaccines. There are many planned or ongoing neoantigen vaccine trials as reported in this review paper, including Mayo Clinic's own trial that you can find on the clinicaltrials.gov website. Currently, we have 2 N=1 compassionate use neoantigen vaccine trials. The first is for a kidney cancer patient. 19 neoantigens were delivered to the patient, and the patient achieved partial response after 4 months with more than 40% of tumor shrinkage at multiple sites. For the first patient, we have the laboratory binding assays of the predicted neoantigens. 14 out of the patient's neoantigens are HLA A0201 binders, arising from SMV, indel, fusion, and splicing mutations. We were able to validate the lab. In the lab, 13 out of the 14 nominated neoantigens, which is a 93% validation rate. We really hope that this new immunotherapy agent is able to help this patient, as we have shown the partial response after 4 months. We also have 2 IRB protocols, one for sample collection, neoantigen identification and validation, and one for a phase 1 clinical trials of solid tumors, which is separate from the 2 individual patient trial. We're also working around the clock to improve the performance of the RealNeo pipeline. For example, to incorporate tumor microenvironment data for patient selection, neoantigen prioritization, and for the therapy outcome prediction. In addition, we are actively exploring vaccine manufacturing options. And the last slide is the acknowledgement slide, and that's the end. Thank you.